Hello, my name's Michael Wilson. I'm an associate professor in the UCSF Weill Institute for Neurosciences in the Department of Neurology. I see patients who have inflammatory conditions of the brain and spinal cord. That includes diseases where the immune system is overactive, like multiple sclerosis, but then also diseases in which infections can cause inflammation of the brain and spinal cord. And my laboratory does research to better diagnose and treat these conditions. The title of my talk today is Solving Neurologic Mysteries, Next Generation Approaches to Diagnosis. As a disclosure, my lab is a member of the UCSF Center for Next Gen Precision Diagnostics, which is a group of labs at UCSF who are launching innovative new diagnostic tests. We don't get any compensation from this, but we're all invested in making this a success. So first, to define our terms. So many of the conditions that we do research on involve inflammation of the brain, the spinal cord, or the coverings of the brain and spinal cord called the meninges. And so when the meninges are inflamed, we call that meningitis. When the brain itself is inflamed, we call that encephalitis. And when the spinal cord has inflammation within it, we call that myelitis. And these are some images here of this is a patient who had a meningitis. You can see these thin coverings around what we call the brainstem have contrast leaking into them because the blood-brain barrier is disrupted because of active inflammation. So this is a picture of meningitis. This is a patient who has encephalitis, and these deep brain structures here are too bright on the MRI, again suggesting that they're inflamed and swollen. So this is a picture of encephalitis. And then this is a patient who picture from the side of their spinal cord, and you can see here that there's some abnormalities within the spinal cord itself, again with contrast agents leaking into it because of active inflammation. And this is a patient who has a disease that's causing myelitis or inflammation of the spinal cord. Sometimes patients can have more than one of these areas inflamed, and so this is a patient who has Meningoencephalitis. So this is someone who has inflammation of the meninges, again, around the brain and brain stem, but then they also have big areas of inflammation within the brain itself. Those terms are fancy terms to describe where the inflammation is, but they don't tell us what the underlying cause is, and that's really going to be the focus today, is trying to talking about new technologies that we've been developing to try and get at the underlying causes in different people. Encephalitis, it's by itself there are about 20,000 cases a year in the United States. Many patients who develop encephalitis become quite sick, and so even though it's a relatively small number of patients compared to diseases like diabetes or heart attack or stroke, these patients are quite costly to take care of. About 10% die in the hospital, and survivors frequently have long-term disability like speech problems, memory problems, or difficulty moving around. A big focus of our research and in the field is this problem of just encephalitis of unknown etiology or encephalitis of unknown cause. And it's a big problem. So about half of cases never get a diagnosis. A long-standing study that was done and led by Dr. Carol Glazer called the California Encephalitis Project enrolled at least a couple of thousand of patients over the years, and despite doing extensive testing, 63% of cases went undiagnosed. So that's a real problem because it makes it impossible sometimes for physicians to employ the best treatments for patients if they don't know what the underlying problem is that they're trying to fix. Part of the reason that we don't do such a great job with diagnosis is because just on the infectious side of the coin, there are so many different causes, and each one of them is relatively rare. So testing for individual infections is hard to come by. There's well over 100 different pathogens that can cause encephalitis, and they vary widely based on where someone lives, the time of year it is. Mosquito-borne infections like West Nile virus are much more common in the summer than they are in the winter. The patient demographics, so some infections people are more likely to get based on certain professions or hobbies. And then there are things like epidemics, which we're experiencing right now, where new infections can sweep through that were just never anticipated before, and for which we don't have focused diagnostic tests. And so that just, again, highlights this emerging infection problem, which again is very timely. There are many emerging infections can invade the brain, some more than others. And I have SARS coronavirus 2 down here at the bottom. There's some early data coming out now that although it's by far and away its biggest, the organs it attacks the most are primarily the lungs and to some degree the heart. There's some early evidence that in rare cases, this virus, that's a respiratory virus, may cause neurologic complications. To highlight how an emerging infection can cause problems for diagnosis, so Zika virus became epidemic in Latin America a few years ago, and a paper that was published in 2016 suggested that Zika had actually been circulating in Brazil for 18 months before it was actually detected. Again, that wasn't because the doctors were not doing their job, but it was rather that Zika virus had only been seen primarily in Africa before. So it just didn't make sense to send off a Zika virus test for patients who were having flu-like illness. So in addition to taking 18 months to just know that it was in Latin America, it took another 6 months or 2 years total before people were recognizing that Zika virus was a cause of fetal brain abnormalities and also causing an autoimmune disease of the peripheral nerves in patients called Guillain-Barré syndrome. Again, that was because one, Zika virus wasn't known to be circulating in Brazil, and also because Zika virus had never been linked with neurologic disease before, so it didn't make sense to look for that as a cause initially. Another reason why we do a poor job at times to make diagnoses in these patients is that although there are many infectious causes of encephalitis, we're now recognizing, as this recent memoir that was published, Brain on Fire, that many cases that we previously thought were infectious causes of encephalitis are actually autoimmune conditions. And this is a story of a young woman who developed a form of encephalitis that was just discovered about 13 years ago. Again, it's an autoimmune disease that used to be confused with a viral infection. It's very treatable. This woman recovered and wrote a book about her experience, but the treatment for it is immune suppression, which is exactly what you don't want to do if you think that someone has an infection. So there's a real tension here when seeing one of these patients in the hospital who are quite ill between trying additional antibiotics or other antimicrobials, or trying to do the opposite and suppress the immune system on the chance that they might have an autoimmune disease. So the type of diagnostic testing that is kind of conventional and is the primary means that we use to diagnose infections really takes advantage of unique aspects of particular viruses or bacteria or fungi or parasites, which is focusing on unique genetic sequences for one virus or another, unique antibodies that are produced to one virus or another, and designing tests specifically to detect those unique aspects of a bug or a response to a bug. And that's what we call a candidate-based approach. meaning that the physician has to interview the patient, look at the MRIs, do a physical exam, and come up with a set of hypotheses about what the person might have, and then send off individual tests for each of those infections to either rule them in or rule them out. And so that process can certainly work, but it's blind to things that the physician doesn't think of, either because it's a rare infection they're not aware of, or maybe it's an infection that no one's aware of, or that no one is aware could be causing the symptoms that are manifesting in that particular patient. So the approach that we've been taking here is what we call a metagenomic approach, where instead of looking at unique aspects of a particular infection, we look at what's shared between us and almost all infectious agents, which is that we all have nucleic acid, meaning we all have a genetic code, either DNA or RNA. And metagenomics is simply just the analysis of all the genetic material in a particular sample. The sample that I get most excited about is spinal fluid. And with the amazing advances in our ability to sequence things very cheaply and quickly, we don't need to go in with probes that are specific for a particular organism. We can just say, let's sequence all the genetic material in a sample and then ask what sequences come out that are not human. And then by doing that, we can try and figure out what the infectious or foreign agent is. And so just to drill down on that a little bit more, so we take a patient sample. This is, again, spinal fluid. Sometimes it can be brain biopsy tissue. We extract the genetic material from that tissue, and then we go through a series of steps to prepare that DNA or RNA for sequencing on one of these tools that are made by various companies that can generate millions of sequences within a day and allow us to do analysis on those sequences. In many cases, we'll get about 10 to 20 million sequences for a given sample. And then the primary goal of the computational side of the equation is, again, to get at the non-human sequences that are present in the sample. And so the first step is we take advantage of the fact that we know the sequence now of the human genome. We match the 20 million sequences up against the human genome, and anything that has a good match gets set aside, because for our purposes, it's not interesting. We want to look at the non-human sequences. So in this case, we go from, we're able, just after a first step, we're able to eliminate 92% of the data. And then we go through a series of additional steps with quality filtering, eliminating just repetitive reads or low complexity reads, meaning sequences that just are strings of single letters like A's or T's. And then we do some more human removal. And in this case, we go from 20 million sequences initially down to just a couple of thousand that we now know are high quality, non-human, and not redundant. And it's those sequences that we then search in large publicly available genetic databases to see what they best match to. And so there's 4 kind of niches where this type of technology can really have an added benefit. So it's able to surprise you in many ways. So you can find novel infections that you're just not aware of that may have a weak match to a microbe that's already present in the database. You may find an infection that people do know about. It's just not known to cause the clinical condition that you're seeing in a particular patient. You may find an infection that's known to cause encephalitis, but it's just so rare that people just aren't widely aware of it. And then, since it's a test that can essentially find any infection, the hope is that a negative result will really help physicians move on to considering other possibilities in that patient, specifically thinking about autoimmune diseases and feeling more comfortable about immune suppression. So here are some early kind of proof of concept cases that we've published on. So this was a case of a young boy in Wisconsin who had was born with an immune condition where he didn't fight infections well. And when he was 14, over a period of months, he had progressive Meningoencephalitis. And although he was extensively evaluated for an infectious condition, because all those candidate-based tests came back negative, the doctors even considered in him that he might have an autoimmune condition. That strategy did not work. And so we looked at his spinal fluid and found he had an unanticipated bacterial infection called neuroleptospirosis that was actually quite treatable with a simple antibiotic. Another case that we worked up was of a woman in her 70s in San Francisco who had developed a pneumonia and then an infection in her eye, followed by a raging infection in her brain. So she had a meningoencephalitis. She had a brain biopsy. She actually had two. And this is a picture of a cross-section of a blood vessel in her brain. And you can see there are these large cells that are ringing the blood vessel and a lot of inflammation around it. So she had a condition that we call vasculitis, so inflammation of the blood vessels. And there was no diagnosis made with traditional testing, but on sequencing, we found a number of sequences that matched to an amoeba. And when we look back at the brain tissue and stain these big blue cells that are present around the blood vessel, they all stain positive for a particular amoebic infection. This was a case of a man, again, who didn't have a normal immune system, who developed encephalitis, so inflammation in one of his temporal lobes, and then recovered without a diagnosis. And 3 years later, this is his MRI early on, and then his MRI 3 years later, you can see that this black area, the ventricle, the fluid-filled space, has gotten much, much larger over a 3-year period. And we investigated him for a possible infection, and he had a rare mosquito-borne virus, Cache Valley virus, that had been causing a smoldering infection in him over a 3-year period. What was interesting, one of the interesting things about this virus was that this man lived in Australia, and the virus had only been seen in primarily in North America. It turned out that when he became sick initially 3.5 years prior, he had just come back from a rafting trip in South Carolina. So we think that he actually, when he came back, he traveled, he brought the virus with him to Australia. We don't have any evidence that it spread beyond him. But again, it just shows that this type of technology can find an infection that's not known to be in a certain part of the world. And then lastly, this was another case of a woman, very healthy 26-year-old who had a chronic meningitis. So she had meningitis lasting for over a year. Sequencing in her identified a rare fungal infection. And this infection is typically seen in intravenous drug users. And that part of the history came out after the infection was diagnosed. This technology has some challenges. So there's just very little genetic material, whether it's human or not, in spinal fluid. We're talking at the level of picograms. So that it's taken a while for the technologies to develop to be able to recover and make sense of those tiny amounts of RNA and DNA. As you can imagine, most of the material that is recovered is human, and so that occupies a lot of the data that you generate. And so the lab I did my postdoc in, Joe DeRisi's lab, developed a CRISPR-based tool, a technique to selectively deplete genetic sequences that are human, that are particularly prevalent in these samples, to be able to kind of gain up the signal on what microbial sequences might be there. A second pitfall when doing this type of analysis is that when we get spinal fluid, we do something called a spinal tap, which involves putting a needle through the skin in the lower back to get spinal fluid. And as the needle passes through the skin, it picks up, even though the skin's been sterilized, it still can pick up just dead genetic material from the skin, from bacteria on the skin. And so when you do sequence spinal fluid, it basically looks like everybody has bacterial meningitis, because you see bacterial and fungal sequences in all these sample sets, because again, you're sequencing everything. So we've done a lot of work to develop analysis tools on the computational side of the algorithm to be able to filter out the expected mostly bacterial and fungal sequences that we think are not relevant to a disease and are not actually present in spinal fluid, but are rather contaminants from the skin and from just reagents that we use in the lab. You will see some papers out there and maybe even some news reports about a brain microbiome, about the possibility that we all have bacteria living in our brain. Look, there's only a handful of these papers, but and I think we're concerned that many of the bacteria that have been reported there are actually these environmental contaminants that are being confused for organisms that live in the brain. We don't have good evidence for that right now. So I'll just go through a little bit more detail on another case. This was most of the time when meningitis or encephalitis occurs, it's acute, meaning it just comes on over a few hours to days, and then it resolves over that period of time as well. But sometimes you can have a really chronic condition. So this was a 40-year-old physician, and she actually had 15 years of relapsing myelitis, so spinal cord inflammation, and arachnoiditis, which is a focused area of meningitis. She'd been evaluated at a number of top academic centers around the country, and her history, she immigrated to the U.S. from India at 22 years old. She lived in Arizona, New York, and Maryland. She didn't have a history of drug use and didn't have any known animal, mosquito, or tick exposures. But then in 2002, she suddenly, over a period of days, developed back pain and malaise. She had night sweats. She lost her appetite. She had a headache and neck stiffness, all symptoms that are common when someone has a meningitis. When they looked at her spinal fluid, instead of having 0 to 5 white blood cells, she had in the hundreds, which is very abnormal. Her protein was elevated and the sugar level in her spinal fluid was decreased. This is all consistent with a meningitis and most suspicious for what we think of as meningitis due to a bacterial infection or mycobacterial infection like tuberculosis. So this was her MRI, which showed some abnormality in the spinal cord and a little bit of that enhancement where the contrast is leaking into the meninges at the bottom part of the spinal cord. So because she's from India, there was a suspicion for tuberculosis, and so she got a partial treatment for that and did improve. But then 4 years later, right after giving birth, she got sick again with the same set of symptoms. She got some antibiotics right away, but never fully recovered, still had these low-grade symptoms for months. She got another spinal tap, which again was very inflamed with a low sugar and a high protein. They even did a biopsy where they opened up the lower part of her spine to take a bit of tissue and just saw, again, inflammation, but no particular organism. So they gave her another year of TB therapy, thinking that was the most likely cause. She got better, but not all the way. She still had low back pain and chills. And then after another medical event in 2015, these same set of symptoms really revved up again. This time they tried treating her for a viral infection and gave her some steroids to just generally bring down inflammation. And for the next 2.5 years, they were not able to get her off the steroids. Whenever they would taper them down, she'd have a flare of her symptoms. and she even had added on chemotherapy agents to keep the inflammation in check. And despite all these immune suppressing medications, she still had relapsing symptoms for the next 2.5, 3 years. So it was at that point that we got contacted to look at her spinal fluid. When they did the spinal tap, she still had low-level inflammation. And when we did sequencing, we found that she didn't have sequences that matched to tuberculosis, but she did have many sequences that matched to a pork tapeworm called Taenia solium, which is the cause of a disease called Cysticercosis. It's actually a common, so Taenia solium, this pork tapeworm, these are the eggs that you can see in uncooked pork here, is a very common cause of brain infections around the world. It's a common cause of epilepsy, of seizures. But despite it being a common diagnosis, it wasn't considered in her case because she didn't have the typical cysts that are seen in the brain on MRI. And so, again, this is a great example of a common infection presenting in an uncommon way. And so it wasn't, the specific test for that organism had not been sent off yet. And the unbiased approach that we took, where we just said what's in there, uncovered this infection that she's now recovered from. So I've told you a lot about a bunch of case examples, and we've done some case series, and that this all built evidence to say that this was a worthwhile approach to pursue, and in collaboration with and really led by Drs. Charles Chu and Steve Miller in lab medicine here, they did a clinical validation of this assay, so which was not trivial. So this normally when you're validating a new test for herpes virus or herpes simplex virus or for syphilis or for HIV, you compare it to how the old test for that infection worked, and you say this new test, works as well or better as the old one. It's a little more complicated to compare a test that can find any infection and to figure out how to benchmark that. And so that's exactly what Charles and Steve did in close consultation with the FDA, and published their validation work just last year. And so once this test was clinically validated, again, this is actually the first time this has been validated for spinal fluid. A study was launched called the PDAID study, so Precision Diagnosis of Acute Infectious Diseases, and it was a multi-center study in the U.S., and the idea was to enroll patients who were in the hospital with meningitis, encephalitis, without a diagnosis. And just to give you the top line results, there were 58 infections identified in these patients. And of those infections identified, 13 were identified by metagenomic sequencing alone. 19 were co-identified by both sequencing and other tests that doctors sent off. And then another 26 were diagnosed by conventional testing. So I'll quickly go through those three buckets. So in the sequencing only infections, 10 had not been entertained by the treating physician or had been missed by traditional testing. And those, again, included some surprising diagnoses, a mosquito-borne virus, St. Louis encephalitis virus, which hadn't been seen in California in 30 years in a human. Hepatitis E virus, which, as the name suggests, normally a cause of liver infection, was identified in a patient. She'd actually had a lung transplant 6 years before and had had mysterious liver inflammation ever since the lung transplant. It turned out that when the hepatitis E virus was treated, it both resolved her meningitis, but it also resolved the impending liver failure that she was suffering from. So those were some of the highlights from the sequencing-only infections. And when we compared sequencing of spinal fluid to other what we call direct detection tests of spinal fluid, like culture or PCR or antigen testing, sequencing performed quite well. But I did tell you there were a number of cases in which sequencing did not make the diagnosis, and rather the diagnosis was made by other types of tests or in other types of tissues. So in 11 cases, serology made the diagnosis, which means that antibody testing was performed. So those are what we call indirect tests, where there's no evidence that the infection itself is present in the spinal fluid, the RNA or DNA, but there are antibodies that the immune system has made against it, and those help with the diagnosis. And we'll talk a lot more about that in a second. Then there were other cases in which the bug was not present in the spinal fluid, but it was present in other tissues, like in an abscess, so the infection was diagnosed there. And then there were 8 cases in which the infection was identified in the spinal fluid, but at such low levels that sequencing picked it up, but it was below what we call the reporting threshold for the test, and it was more sensitively picked up by another type of test, like a PCR. And so this leads into the other kind of the side of the coin of what we've worked on. So we know that direct detection tests, like where you're looking for the bug itself or its genetic material, can be quite successful, but they have some disadvantages. So many neurologic infections can only stick around in the spinal fluid for hours to a few days. And so if you do a spinal tap not at quite the right moment, you may miss that microbe and miss the opportunity for making a diagnosis that way. And as I mentioned too, sometimes these infections are compartmentalized. So a patient may develop an abscess and they may not be leaking much of the microbe into the spinal fluid. And so you can miss an infection by only testing spinal fluid. And so there's a number of viruses that that can cause encephalitis that we actually don't typically diagnose with direct detection testing. Those include West Nile virus and a number of other rare viruses, but even common ones that we think about, like rabies or the chickenpox virus, varicella zoster. Those are diagnosed typically by antibody testing. And so we've been wanting to complement the sequencing approach to diagnosis with an antibody approach that's also very comprehensive and doesn't require kind of having a hypothesis about what the person might have up front. And so in collaboration with Joe DeRisi, we've been working for a number of years now to develop a sequencing compatible, cheap, plate-based, robot-friendly, and scalable technology to really broadly look for antibodies against a number of viruses. The technology we built upon was first developed by the ELIGE lab at Harvard. And so this work was really spearheaded by Akshaya Ramesh and Ryan Schubert, who were postdoctoral fellows working with me. And they built a library of genetic sequences, about 500,000 of them, that tile across all vertebrate, tick, and mosquito virus genomes. And we ordered those sequences to be, we ordered them from a company to be synthesized. And then we take those about 500,000 short pieces of DNA, and those get stuck into another virus's genome, so something called a bacteriophage, which is a harmless virus that infects bacteria, and we can manipulate its genome. And so these bacteriophage, which look a bit like lunar landers, we can insert a little piece of another virus's genome into this phage, and specifically into a region of its genome, so that that little piece of DNA will get turned into a peptide, a protein, and expressed on the surface of that virus. So this, you could imagine, is a phage that's expressing a little bit of a piece of protein from West Nile virus or from SARS coronavirus. And we can take that library of phage that each express a little piece of another virus on its surface and combine it with a patient sample that has antibodies, and just ask the question of which phage bind to the antibodies that are present in the patient sample. We can wash away the phage that are not bound and then sequence the genome of those phage that are bound and just ask which viral peptides did the person's antibodies pull out. And so we applied this technology to an illness that's been making the news over the last few years. So this is something called acute flaccid myelitis, or a polio-like illness that has been spiking in incidence since 2012, every other year in the U.S. These are children who are typically quite young, usually under 5 years old, who get a respiratory illness and then a few days later suddenly become limp in one or more limbs. And this is how we used to think of how polio presents, but polio isn't present in the United States anymore. And so people have been searching for another potential cause. There's the leading suspicion has been for a cousin of polio, another enterovirus. But because we and others have not been able to detect the enterovirus directly in spinal fluid from these children, there's been a concern that there may be some other virus that's out there that's being missed, at least in some of these children. And so we teamed up with the CDC, with the Chan Zuckerberg Biohub, and California Department of Public Health, and a number of academic centers to collect spinal fluid from children with AFM, with this polio-like illness. And we also have spinal fluid from kids with other neurologic diseases. And we basically ran this assay. So we incubated the spinal fluid from each of these children with this phage display assay that has peptides, proteins represented from thousands of different viruses. And we just said, in the children with this polio-like illness, what viral antibodies do they have in their spinal fluid that the kids without this illness don't have? And so the only viral family that was enriched in the kids with this polio-like illness were the picornaviridae, picornavirus, and specifically enteroviruses. So these children clearly had high levels of antibodies to enterovirus in their spinal fluid, whether they'd had enterovirus detected or not by PCR or by sequencing. So it really lent additional weight to this initial suspicion that enteroviruses really are responsible for this illness. And this technique also allows us to document where along, this is an enterovirus genome, the different proteins that make it up, and we can show specifically where along the genome of the virus patients are making antibodies to. And this work was also supported by another publication that came out right around the same time. Okay, so now I want to shift gears and tell you one last chapter here. So I've mentioned along the way that some patients certainly have infectious causes of encephalitis and meningitis, but we also recognize that a number of patients who we think have an infectious cause of brain inflammation actually don't. And so we've been, again, working to develop broad-based tests to look for autoantibodies, so antibodies that target our brain tissue or spinal cord tissue and cause these diseases. And a recent study actually suggests that autoimmune causes of encephalitis are at least as common as infectious causes. And there's a number of these now. There's at least 20 to 30. The number of antibodies that we know can cause these diseases keeps growing. These are just 3 of them here. And the traditional methods for finding these antibodies, one is to take a brain slice from a rodent like a mouse or a rat, and to see whether the antibodies in human spinal fluid bind to anything in that brain tissue, suggesting that there's some attack of a brain protein. That technology is helpful. It tells you that there's probably something going on, but it doesn't tell you exactly what the protein is that might be targeted. There's a powerful but kind of low throughput method called immunoprecipitation mass spectrometry, where you can try and identify the specific proteins, and we definitely do that. But again, it's a low throughput technology, and so again, we've taken a phage-based approach, and the kind of overall name for this technology is programmable phage display. Another term that's used is PhipSeq, or phage immunoprecipitation sequencing, which was initially developed by a laboratory at Harvard. And taking a page from their work, Brian O'Donovan, shown here as a graduate student, designed another phage library in which instead of designing viral proteins to be expressed on the surface of these viruses, he designed a library in which pieces of human proteins are expressed on the surface. And so I'll tell you about a case in which we used that technology. So this was a patient that I saw in clinic a few years ago. He's a 37-year-old right-handed man. He'd had a history of testicular cancer a few years before, but had done very well. And when I first saw him, he'd had 9 months of vertigo that worsened, and about 5 months before I saw him, and it had been complicated by constant double vision. He felt like he was constantly walking in a canoe. He had a tremor in his right hand, and these symptoms were so bad that he'd had to stop driving, he'd had to stop working. He had twin infants at the time, and he couldn't bend over to pick up his children without falling over. He'd had a spinal tap, which also had inflammation, so the number of cells was increased, not to the same degree that we saw in the chronic meningitis patient, but definitely not normal. And he also had evidence that he was making extra antibodies in his spinal fluid, and the question was, what were those targeting? He had a brain MRI, and it was largely normal, except for a small area of abnormality in an area called the brainstem and cerebellum. And this is exactly the location of the brain where if you have inflammation, you will get significant imbalance and eye movement problems. So the location fit with the symptoms that he was having. So because of his cancer history, because of he didn't have fevers, and there were other tests that suggested he didn't have an infection, there was a worry that he might have one of these autoimmune conditions, and some of these can be stimulated by a history of cancer. And so we attempted to just right away suppress his immune system with steroids. He got initially better, but then had to transfer his care because he had lost insurance, and over the next year was given multiple other diagnoses, including multiple sclerosis, and then a concern when he didn't improve and actually continued to get worse, that he might actually have a brain tumor. And this is a picture of his MRI about a year later, and you can see again in this brainstem area that on one side of the brainstem is slightly larger than the other, and when things get bigger in the brain, we worry about tumors. It turned out this was not a tumor. This was a consequence of just chronic damage to this part of the brain. And actually, this was due to inflammation. And so while he had transferred to a different insurance, we were still working on his spinal fluid in the lab, and this is what we found. So this was work, again, started by Brian, and then really spearheaded by Callie Mandelbrem, and in collaboration with Joe DeRisi. Using this phage technique, there was a clear antigen or target of the antibodies in his spinal fluid, which is some protein called KLH11 protein 11, and we confirmed this in a number of ways. And what was interesting was, we were very excited to find that this protein in this patient, but we didn't know how significant this was beyond his particular case. Was he going to be a snowflake, a unique case, or was this going to have implications for other patients with these kinds of diseases? So with some serendipity and collaboration with Sean Piddock and Div Dubé at the Mayo Clinic, it turned out that they had been collecting patients who also had a history of testicular cancer, who also had a history of severe imbalance and vision changes for about 20 years. And all these patients had a very typical staining pattern on the brain slice that I mentioned earlier. So that it was actually called sparkles. The proteins that lit up kind of appeared like they were stars in the night sky. And so they've been collecting cases like this for years, but did not know the actual protein being targeted. So we told them about this KLH-like protein 11, and they looked to see in their cases whether that was the target in their men with these symptoms, and it turned out they all had the same target. So now instead of this being an antibody associated with a disease in one person, is associated with a disease in 13, and now it's well over 50 patients with this. So we think this is a second autoimmune cause of encephalitis in men with testicular cancer, and as these diseases go, it's a rare disease, but not uncommon for these types of conditions. So just to conclude, on the sequencing side, this is a test that's now clinically available at UCSF. It's, to my knowledge, UCSF is still the only place in the country with a clinically validated metagenomic test for spinal fluid. We, as a large group, have shown that CSF sequencing increases diagnostic yield in patients with meningitis and encephalitis. And some things that I didn't get into more detail on is that in addition to finding an infection, because we can sometimes recover the whole genome of a particular virus, we can get a lot more information beyond just saying, oh, there's this infection there. We can track, we can make guesses as to where the particular virus came from, whether it has, or a bacterium, what particular resistance mutations it might have to certain antibiotics, and a number of other analyses that can be done downstream. I also told you that sequencing, like any test, is not perfect. There are cases in which it might miss something, and so that's why we've been hard at work at trying to develop these Plan B type tests, these antibody tests, to be also able to find viral infections and even new autoimmune diseases. Phage display is one technique that I've told you about. It's very exciting. It's not perfect, and so with this technology, we're looking at pieces of proteins. Sometimes these antibodies really need to have a whole natively folded protein to be able to be pulled out. And so there's additional work going on now to complement the phage display technologies to find additional antibody targets. And I just want to acknowledge that this work is a result of many groups at UCSF, my lab, Joe DeRisi's lab, Sam Pleasure is also in neurology, does a lot of the brain slice staining with us. Charles Chu and Steve Miller have been real pioneers in this effort, and we've been very fortunate to get a lot of philanthropic and governmental support for this work. So thank you.